Hello, everyone. Thank you all for being here at our webinar about food safety and security. I'm happy to see you. And I will start with a short introduction to the event and walk you through the schedule and who we've invited today. And then, we can start with the speakers' presentations. Okay, so by 2050, feeding 9 billion people without depleting Earth's resources is a major challenge. And our current food production systems also increase the potential for spillover of pathogens because of agricultural expansion and that's into wild habitats. And this increases interactions between wild animals and livestock or humans. And also there's the intensive farming practices. And in that way, pathogens, which are also sometimes antibiotic resistant, can spread rapidly among animals. So this requires an educated and skilled workforce to address food-related issues. And this includes experts from different fields. And adopting a collaborative One Health approach will be crucial for effective problem solving and mitigating spillover events and other effects of food production. I just muted it. Now you should be able to hear me again. So since the One Health approach is essential for addressing food safety and security concerns while feeding the global population, then what is needed and what are the benefits? So key aspects of food safety, such as education, sanitation, hygiene, food science, public health, and good manufacturing and agricultural practices, they remain essential. But the recent food security and safety issues show that solving specific risks often requires interdisciplinary approaches involving experts from various fields, such as veterinarians, wildlife specialists, epidemiologists, toxicologists, and microbiologists. And then in that way, a One Health strategy can help reduce food waste and boost nutrient utilization through closed circular food systems, and also help mitigate emerging zoonotic diseases and antimicrobial resistance. So to implement this One Health approach, we need to improve collaboration between veterinary, medical, environmental, and legal professionals. And the key actions include promoting sustainable farming, strengthening surveillance and response systems, and aligning food safety laws with global standards. And then public education programs are vital for raising awareness about safe food handling, the impact of dietary choices on health and the environment, and the importance of reducing antibiotic use in agriculture to combat this antimicrobial resistance. So the webinar of today aims to provide an overview of One Health and the approach in food safety and security. We will look at the importance of interdisciplinary collaboration and public education through case studies by our speakers. So the introduction, we will allow some time for people to drop in, and we have some time left. So I just kind of walked you through the standards concepts we will be discussing today. And then Akbar will start his presentation. And you can ask him questions in the Q&A. And after this, there's a slight change in plan. So instead of Benny Osborne, Sara Garcia will give a presentation, while Osborne will still give a short introduction to her and share some opportunities. So Akbar Ganatra is our first speaker. He's a medical entomologist and parasitologist at the Laboratory of Entomology here at Wageningen University. He started his studies at Kiel University in England, and his research there focused on genetics and entomology, mainly on the Anopheles gambiae, and this is a deadly mosquito species that is prevalent in his home country, Kenya. And then he did a PhD, which was on the impact of agrochemical pollution on schistosomiasis infection and transmission in Western Kenya. And he will dive deeper into this in today's webinar. So currently he's a researcher at WUR and also involved in many organizations focused on environmental conservation, global health, and collaborative research. And then our second speaker, Sara Garcia. She coordinates technical assistance activities for climate smart agriculture, which is an incentive program at the Californian Department of Food and Agriculture. She previously worked at UC Davis, the Western Institute for Food Safety and Security, so like Benny Osborne, and here she developed education, outreach, and technical assistance programs for agricultural producers. And she also earned her PhD, which was in integrative pathobiology at UC Davis, and she completed her postdoc in Rwanda, where she supported capacity building in dairy production and food safety. So I hope you enjoyed today's presentations. We will start with the presentation by Akbar, if you're ready.
Yeah. Oops. That is a spoiler. Yes, so I am Akbar Ganatra, and as I was presented, this was for my PhD, and it's the effect of agrochemical pollutants on transmission of schistosomiasis in Western Kenya. So schistosomiasis is a neglected tropical disease. It affects around 200 million people currently infected, and kills around 300,000 depending on the estimates you kind of look at. So it's kind of like a slow killer. It's kind of neglected because the deaths go unaccounted for. It's caused by comorbidities. So it's not really usually you can pinpoint that the death was caused by schistosomiasis. So nowadays they use a different measure to kind of measure the effect of this disease, which is the disability adjusted life years. So this is how many hours are lost, from work or school due to the disease. For example, if the child has anemia and cannot play, how much of the development is affected. So this is currently estimated at about 3.3 million disability adjusted life years. It's mostly an African problem, as you can see. So 90% of infections and mortalities happen in Africa, but it does occur across the world. So there's cases it's prevalent in South America, Middle East, China, and parts of India. It's more, there's two main varieties in Kenya, which is what I'm going to research. So biology of schistosomiasis, since I'm told there's not so many experts, I'll go through all of it. We'll start with an infected person. They usually have a male and female living in copula, which is like the male and the female lives inside the males, and they basically drink blood and lay eggs throughout their life. And this can be about 3 to 10 years. And they lay lots of eggs, which they're hoping would be excreted with your urine or feces. So if we look at, can I do the laser? Yeah. So if we look at the eggs, you can see that they're different shapes for the different species. They basically, once they enter the water, they hatch and they have to seek out a snail. So depending on which species will be which snail. And they have 24 hours to do this. So this the Miracidia seeks out a snail, and once it catches the snail, it undergoes asexual reproduction. So they turn into sporocysts and they reproduce asexually, and this takes about 4 weeks. And then they come out as free-swimming cercariae. So these are the human infective stage. So like the Miracidia, they only have about 24 hours of energy in the water, after which they die. And during which they're just seeking out, through the smell of humans and maybe the wave action, they find the human and they can penetrate the skin. So you don't have to swallow it, you can just be standing in the water. Once inside the body, they use their circulation system to find your liver, and there they mature, and then they move to their preferred location in the body to mate, which is, depending on the species, either the intestines or the bladder. This is where I began, where they're just sucking your blood and laying lots of eggs. So The disease primarily comes from these eggs. Instead of leaving the body, they get reabsorbed into the circulatory system, and then they get stuck in places in the body they're not supposed to. Because your body doesn't really have a defense for them, all you can do is just trap them in like scar tissue. So granulomas form, and so if the eggs, they mostly get stuck in the liver, they get stuck in the liver, and then the body just covers it in granulomas. And then that's what the picture you saw in the beginning, where it was the hepatomegaly and splenomegaly. The liver becomes enlarged and it's just covered in scar tissue. So it really stops being useful as well. And this is where the disease symptoms comes from. But these eggs can get stuck virtually anywhere in the body. About 4% of cases are schistosomiasis, where they get stuck in the brain. And there's a case study where they got stuck in a child's spinal cord and he lost the use of his legs. But they can be removed. So they can operate and remove the scar tissue, of course, if they find it in time or if the person can afford it. And treatment is actually quite easy as well. So if you have the disease, you can kill these worms with just praziquantel, or it's basically a deworming tablet. And you just have a few of these. The problem is it only kills the adult worms. So if you have these immature forms, which are taking 6 weeks in your body before they turn adult, then that medicine did not kill them. So if you have the medicine, you can kill the worm, but then you need to have it again after 6 weeks to kill all the immature forms that have now turned into the adult forms. So this is basically kind of easy for people who are not in endemic areas, because then they can make sure that their body is rid of this. But then for a person in the endemic area, unless they're taking the deworming tablet every 6 weeks, they have to make sure that they're not getting infected as well. Okay, that is, I think, everything about the biology. So agrochemicals, are they now a new problem for the disease? So agrochemicals like pesticides, we have the two sides of the argument. One side saying that, hey, we have a growing population and, we have to feed this population. It's already so hard as it is with all the challenges that we have, pests, climate change now as well. So pesticides are really our one, armament. At least this is the argument. On the other hand, there's, well, I use this image just because it kind of shows all the different ways that pesticides impact. Not necessarily that it has a financial impact, but this just shows that the pesticides really do have a negative impact in a lot of different areas. So they get into, oceans, fisheries are affected, obviously, even our rivers and things, groundwater, which is a problem for humans. There's bird loss, our domestic animals are affected, unintended crop losses. There's already direct health impacts for pesticides, so cancers and things. So in this thesis, I basically talk about indirect effects. So that's not even been accounted for. And then there's of course the things that we've not even yet discovered. So there's a lot of things that pesticides already affect. And basically in Germany, they already knew this. So they started collecting, water samples and checking for pesticides a long time ago. But then they realized that it's quite expensive and time consuming. So they were in the they were basically building this biomonitoring biomonitoring kind of system they call species at risk. So this one, it's basically, it tells you instantly the level of pollution in a river by looking at the community composition of the river. So naturally, some species are more sensitive to pesticides and other pollutants than others. So if you go to a river and you find the sensitive organisms, then you can kind of assume that there's no pesticide pollution. Otherwise, they would be dead. On the other hand, if there's some that are really tolerant, and if you only find those in a river, then you can assume that there's been a pesticide pollution kind of event here, because the sensitive organisms have died. So if you look at kind of the ratio of sensitive to non-sensitive, you can reasonably estimate the pesticides pollution. So to do this, they actually had to develop a scale where they test each organism and the sensitivity to pesticides. And it was in doing this that they found out that Planobidae, which is the hosts of the disease, are actually one of the most tolerant to organic and other pollution than everything else. So if I can digress here for a moment, because we were talking about interdisciplinary, discussions. I already had problems in my university because this PhD was too interdisciplinary, and I didn't fall within, the categories already placed. But the argument here, I would say, is the parasitologists would be so busy looking at parasitology and trying to, attack this disease head on and maybe not make progress because, on the other hand, this is going on in the background and they would never notice the link. So I would say this is another argument for actually this interdisciplinary kind of connections and information passing through. Anyway, getting back to it. Planobidae are the most tolerant, or one of the most tolerant. This is them, they are the host snails, to pesticide pollution. So this is where the hypothesis began. Since these are so tolerant to pesticide and other pollution, if these events enter, the pesticide pollution event enters the water, how does that affect the community? Would there be an increase in this tolerant species? So those are that was the background I was working with. And so I began with laboratory experimentation, where I basically replicated the experiment that you just saw before, trying to find the sensitivity of different organisms, because they, of course, started in Germany and I was working in Kenya. So that's us, that's measuring pesticides, and then basically putting oviposition cups with pesticide water, catching insects and seeing what concentration they die. So I'll go right into the results for this. These are the different taxa we caught. So there's like the general batted hemipteran, like water skimmers, Corexidae, water boatmen, back swimmers. Then there's like Ephemeroptera, which are the mayflies. We had Odonata, which are the damselflies, and some beetles. So what's important here is that everything else basically we could kill at low concentrations. Obviously, there's increasing concentrations, but what's interesting to note is these snails are the Planobidae snails, which cause the disease, and we could not kill them no matter how high the pesticide concentration we could get. So I could not get an LC50 for these two species, whereas everything else we could kill, sometimes at very low concentrations. So that kind of gives the first bit of evidence. And this is imidacloprid, which is a neonicotinoid. And I did this for diazinon as well, which is an organophosphate. So in this case, I was able to kill Bulinus africanus, but it was, again, as you can see, the most tolerant species by a lot to everything else. So the lab experiment confirms that these species are more tolerant to other taxa in these waters. So then I went to the field. This is the outline of Kenya. So focusing on Western Kenya. The blue is the lake, and these are the 4 counties I was working on. So this is Migori, Homa Bay, Kisumu, and Kericho. And this is because Migori had sugarcane plantations, Kisumu had rice plantations, and Kericho had these tea plantations. So these were all commercial plantations. And here in the center was mostly maize and mostly subsistence. So I tried to get a mix between subsistence, commercial, and the different farming types as well. So this was some previous literature which showed that both forms of the disease were here. So there's Schistosoma hematobium and Schistosoma mansoni, which is basically urinary, and this one is intestinal disease, schistosomiasis. And you don't need to look at this like too carefully. All I wanted to show was that there was instances of the disease. So there's high levels of commercial farming and the disease. So this was the perfect place to really study it. So yeah, I selected 50 sites all over this area. I went and collected snails and all the other insects I could catch. So that one in particular is a snail catcher, but I did use a sweep net as well. And that's just me trying to pick out and identify things. And this is a typical catch of what you find. So the mayfly, the really small ones, the damselflies, and then there is a snail here, but it's not planorbit. But anyway, this is basically a typical one. There are like hemipterans littered all over as well. So we also collected the water physical-chemical parameters from the water, and we also collected water samples, which were then shipped to Germany. So that's my project partner who helped analyze them via GCMS and LCMS. And then converted this to TU for us. So TU max is basically a standardized unit. You can compare the toxicity of two different sites. So essentially, because there's going to be a whole list of compounds you find at every site, it's difficult to compare, how toxic it is one site to another. So what they did is they compare the how toxic would that water be to a reference organism, and this is what the TU max is. I'll explain it later in some other sites, in some other slides. This is just a list of everything we found in the water, and you can see that we found things in different concentrations, and they belong to different classes as well. So there's a whole list of compounds. I'll put a link, so in case anyone is interested, you can go read the papers in more detail if anything interests you. But to the juicy results, so we found that snails prefer polluted waters in that they were only found in at least moderately polluted waters or greater. So, well, I guess I have to explain to you already. TU is here. So essentially, a TU max of 0 would be that water can kill 50% of your reference organism in 24 hours. -1 is 1/10 of that. -2 is 1/10 of -1, and so forth. -5 basically means negligible or non-detectable pollution. So 0 is again very, very high. So -3 is about moderate pollution. We only found the snails where there was at least moderate pollution. Then to check the independent effects. So some of these things we already knew have an effect on the presence of snails, such as turbidity and dissolved oxygen. So, and dominance of competitors, I think, as well. So it's quite, this is the first fieldwork that showed that pesticide pollution does have a direct independent effect on the presence of snails. So we took those factors and we put them in a hurdle model, and then we put that in a PCA. And basically, this is what I came out with. So again, what's important, well, we already knew that pesticides and turbidity are correlated because when runoff events happen, they kick up the dust, which increases turbidity, but they also wash off the pesticides from the crops. So this is why these two are related. And what we can see from this graph and what our research showed was that when this runoff event happens, the number of competitors reduces. What we did then was we broke down the community composition of the different sites, and we differentiated the sites by the level of pollution. So low versus medium versus high. And you can see here, this is the grazers and the predators. So there's not much difference actually in the community composition between the different levels of pollution. But what we did see was in the high pollution, the grazers specifically become dominated with host snails. So this guild basically is reduced, the dominance of the snails increases over the competitors. So there's less of anything else that feeds on vegetation except the snails. Okay, yeah, so all that was how the pesticides affect the host. So, the lab experiment showed the host snails are more tolerant to everything else, and the field experiment shows they only appear in moderately polluted waters or greater. But what does this mean for the disease? Because the parasite is also in the picture. So I did 3 different experiments to see how do pesticides affect the parasite. I'll go through them very quickly because the results are not that interesting. But first experiment was just to see how the free living stages, which is the Miracidia and Saccharine, react to pesticides. So just put us, I couldn't find pictures of both that would fit well in this animation. So this is an egg, but you can imagine it was a Saccharine or a Miracidia. And put pesticides in it and just leave it there for 24 hours. And yeah, well, I'll come to the results in a minute, but yeah, that's the first simple experiment. The second one is to see how the Miracidia host seeking is affected. So in this one, I put a Miracidia in the water, I put pesticides, and I left it there for 6 hours. So these are the first 6 hours of the Miracidia's life. After that, I give it a snail, and I left it there, I think, for another 6 hours. And I see if that Miracidia was able to penetrate that snail, of course, with different concentrations of pesticides. And then the third experiment is, so this is after the Miracidia is already inside the snail. Since that snail is now being affected by pesticides, would the sporocysts continue to develop normally? So I put a snail in the water, I put a Miracidia in there, I basically let it infect it immediately. So I give them 6 hours to infect it. After that, I give, that Miracidia more time. I leave the snail alone so that the Miracidia has time to go wherever it's going inside the body. And then I put that snail in pesticide water for 24 hours. So then I put it in fresh water for 6 days, and I keep doing this, moving it into pesticides for 24 hours every week. And I do that for 4 weeks. So this snail is basically going through pesticide pollution weekly. And then after 4 weeks, I start checking it for infection to see if it starts shedding Saccharine. And that also goes on for another 4 weeks. So the results, again, I won't dwell too much on it, on the exact numbers, but yeah, you can kill the parasite, but the concentrations needed are much higher than you can find in the field or you would want in the field. Yeah, and of course, these Saccharine die naturally anyway within 24 hours. So I don't know if you would want that payoff, that higher pesticides for killing something that's already going to die. No. I was able to stop the Miracidia from finding the snail at high concentrations, but again, this was very, very high concentrations that you don't want and that you're never going to find naturally in the field. At least you'd hope not to. And for this last experiment, yeah, that was not so interesting. I wasn't able to affect the shedding at all, so they kept shedding. So yeah, that's the conclusions from my talk. Again, the snails from the lab experiment, we could not kill them no matter how high the concentration we could get. So they are more tolerant than all the other taxa. In the field, the snails are only found in at least moderately polluted sites or greater. And while the community composition is not altered too much, the grazer guild does become dominated by snails. And of course, the last experiments showed that the schistosoma parasite would not be affected at environmentally relevant concentrations. So yeah, if I can spend a moment on my acknowledgements. So the DFG, which is the German Science Grant Fund, and of course, Egerton University, CPE, UFZ, Kemri, and these are ICP's core donors. And yeah, you can find more of my papers here. I think I'm ready to take any questions.
Yeah, thank you for your presentation. It was really interesting to hear, like, not just the theoretical framework, but also, yeah, the mechanism of how it could actually, yeah, this pesticide affected these snails that are the host for schistosomiasis, but also then on the other end, how the pesticides don't, yeah, necessarily affect the parasite itself. So yeah, thank you for going into detail about that and the setup as well. I like the visuals. It helps me, yeah, get the methods in my head. So I'm curious if there are any questions from the audience. You can raise your hand, and otherwise I do have a question in the meantime. So you were talking about how, yeah, you might actually felt like your PhD was too interdisciplinary, but that like parasitologists also need other views. And I was wondering, like, what are the major things you like hurdles you encountered in this PhD being too interdisciplinary? Like, was it hard to find supervisors to, yeah, who are knowledgeable enough to help you with all aspects, or was it something else?
Yeah, so this particular case, it was a scholarship, so they had already decided a lot of things. So I was lucky in that sense. But when I was coming to my university, so again, you could see there's a lot of ecotoxicology here. And the main PI, the person who, as I said, was making the spear and she was doing the toxicity sensitivities of the different organisms. Yeah, he was an ecotoxicologist, so there was a lot of this. But the university that I was given was a Kenyan university where they didn't have ecotoxicology as like a course. Of course, there's also parasitology in this. So I was enrolled under parasitology, but then half my papers were ecotoxicology. And so I had basically a problem graduating a little bit because they were like, oh, you have not done enough work for a parasitology degree because half your stuff is ecotoxicology. And then that's actually, I'm going too far ahead. The other problem was even getting enrolled because when I was doing the proposal, they were like, you've spoken a lot about ecotoxicology and not enough about this. But I had to, of course, explain how this was coming about. So yeah, there was the friction at the beginning trying to get accepted into the university, and then there was friction trying to graduate from the university. And yeah.
Okay. Yeah. So it was kind of hard to convince them how important that interface is between like ecotoxicology and parasitology. Okay.
Yeah.
Yeah. Okay. Thank you for your answer. I don't know if anyone has a question from the audience. Oh, I have a question in the chat. So can you compare the levels of pesticides found in the different locations in your project with other locations in the world?
Yes, because, well, if you, I don't know if that QR code is still visible. So there is, my project partners' papers are out there, and you can find either if you want all the information in, I think, milligrams per liter, but then also in TU. So if you want to easily understand it, it's also available in TU that you can, and this is, well, it's not a standard, but I think the biomonitoring kind of application is being, at least when I was doing it, it's well developed in Germany and people had used it in South America and shown that it works. So yeah, I think it's very comparable in different ways.
Yeah. So the concentrations are quite standardized overall.
Mm.
Yeah. Okay. I see a hand by Fatima. Feel free to unmute and yeah, ask.
Thank you. Thank you very much for your presentation. I really enjoyed it because it's kind of my field too. But I would like to find out and also make a comment. I know the snail species, uh, we have different snail species and the kind of schistosoma species that they are infected with. I mean, the host, I'm talking about the hosts in the contaminated water or the environment.
Yes.
But I kind of want to find out, I only heard of Schistosoma mansoni. Was anything done on any other species like bovis or hematobium? Because I know they are all transmitted from the same environment, but different kind of, snail species.
Right.
Thank you.
So, yeah, when I started, at least the toxicity tests with the insects and the snails, I did use both Bamphalaria and Bulinus. So Bamphalaria hosts the Schistosoma mansoni and Bulinus hosts Schistosoma hematobium. So those experiments,
Hematobium.
yes. So those experiments, I did use both the snails. When it came time to actually do the experimentation on the parasite itself, there were a number of challenges. So COVID hit, and in this Western Kenya, there's a lot of Schistosoma mansoni. Like there was reports of both occurring, but I was isolated in that small island. I don't know if my laser is still working. Yeah, this part where there was not so much. So that time there was a lockdown, and that area did not have so much Bulinus or Schistosoma hematobium. So the last experiments I only did on Schistosoma mansoni, but the host experiments I did on both snail species.
Oh, okay. Thank you very much. Okay.
Yeah, thank you for
Can
your
I
answer.
answer?
Oh, yeah,
Yes.
go ahead.
Yes, I want to make a comment on the pesticide. It's very interesting to know that only moderate to higher concentrations of pesticides can affect schistosoma species. The reason why I'm saying this is that as part of the schistosoma control program that has been done by the WHO over the years, attacking the snow species in its environment was one of the methods. So this study is very intriguing to find out that it's only moderate to higher concentrations. And I'm sure this is the reason why the disease is persistent, especially in Africa. Despite the long years of treatment that has gone on in the continent, we are still recording higher and an alarming incidence of the infection. So it's nice to know. So any other further questions, probably I'll contact you later personally so that we can discuss. Thank you.
Yeah, thank you. Although, yeah, I would say that going into this, it was already known that snails prefer a little bit of polluted water. It wasn't known so much about pesticide polluted water, but general pollution, yes.
Yeah, thank you for the addition, Fatima. That's very interesting to see a
Okay.
little discussion. If you have anything to add, of course, feel free.
I'm okay for now. Maybe later on, as we go through this, I will contact him personally if I need further.
Sure.
Yeah.
Sure.
Thank you.
Yeah, thank you. And then I also have a question in the chat. It says, how is your study associated with the One Health approach? And I guess that's mostly because it was on an interdisciplinary interface.
Well, I would say, well, maybe I've not understood the question as well, because it says, how is it associated with the One Health approach? Okay. Well, I would say it's associated because basically the pesticides are a plant health thing. So we put the pesticides there because we want to really take care of our plants. And schistosoma is a human health problem. Of course, you heard about Schistosoma bovis from Fatima, which is an animal health problem. Together, they can also be a zoonotic problem.
Yes, sure.
Yeah, so I think this is one of the few completely, One Health topics out there, because really it's going to take interdisciplinary thinking. Whatever new pesticides or whatever they come up with, we now have this in our arsenal that we have to look at how it affects animal and human health because of such and such case study. We're no longer living in that ignorant past where we have to just work in isolated, like it is a multidisciplinary thing. Similarly, I keep getting told, we can cure this disease by adding molluscicides to the water. And I get told this after I've done this presentation. So I feel like definitely people still don't understand the One Health scenario, because this whole presentation is about the unintended consequences of just, thinking about the one problem and, in sort of most cases, just applying this pesticide or whatever. And yeah, I think we really have to look at, from a bigger perspective at all these areas. How are they interrelated? How are they interconnected? How are they going to interfere with each other once you try to mess with elements of this? But yeah, I think generally the pesticides as well are known to have environmental effects anyway. So there's and human health and plant health, environmental health, animal health, it's all One Health in this project.
Yeah, thank you. I think it was a very clear answer. So I guess indeed, it's not just how to cure the disease, but also the unintended consequences, like of the whole mechanism behind how this disease might be transmitted and what might increase it. And then the effect of pesticides, not just on schistosomiasis, but yeah, of course, there's so many other effects. So thank you for the background. Then I have one more question. So how is schistosomiasis controlled in an endemic area?
How is it controlled? Currently, just through mass drug administration. So there are some countries that have managed to eliminate it through that, but then you have to be very consistent with it. So I think I mentioned it already that once you take the drug, you're still susceptible to the larval forms inside you, and it doesn't actually protect you. So if you enter the water on the same day you had the drug, you will still get the disease 6 weeks later. So I think it was Egypt that upped their mass drug administration up to 4 times a year. A lot of countries struggle. So Kenya itself, the area I was in, they were supposed to be doing it annually, but they weren't keeping up the schedules. And then there's obviously political kind of things. So there's that. And then if you really, really want it, I believe it's possible. And well, there's some research already that shows that in addition to mass drug administration, you do what's called WASH, which is water sanitation and hygiene. So if you implement WASH at the same time, you can prevent people from being reinfected. And that way you can basically eliminate the disease. And yeah. Snail control is also kind of told, but again, if it's by molluscicides, I would not advise it, because again, there might be unintended consequences, which we don't want to go through this entire thing again.
Yeah, exactly. And then, but these snails do prefer polluted waters, right? So they are less prevalent in non-polluted waters.
So that link is basically because it's kind of like a confounding factor. So the polluted waters, or like the really clean waters, happen where the rivers kind of start. So that's where the river is, from what's it called, a spring? Yeah, the spring, and then it's like a headwater stream. Those tend to be very clean, but they also tend to be very fast moving, which is also something the snails don't like. So it could be that the snails don't really like the fast-moving streams, and they end up in the slow-moving things where there's a lot of pollution. But I think with this research, we at least show that they have an additional benefit where there's a lot of pesticide pollution, which is that the competitors are not as tolerant to this pollution. There's also additional kind of, the pesticide pollution also comes with other agrochemicals like fertilizers, which kind of increase the vegetative matter in the water. And that provides an additional food source because it helps the algae grow. Yeah. So yeah, there's more research out there that shows there's other agrochemicals that can also affect this ecosystem.
Yeah, exactly. Okay. Then we move on, I think, to one of the last questions from this Q&A. It's by Tigin. And he asks, what are the potential environmental risk factors contributing to the occurrence of schistosomiasis?
Environmental risk factors. Well, basically is access to clean water. Like if that's one of the risk factors. So if you have to go to a river to get your water, that's one of them. Then, well, income level on some level, but then or age is another thing. So mostly children get it because, again, they're also more likely to be swimming in rivers and things like that. Although at about age 15 or so, the people older than that might have gained immunity at some point as well. So that age is another thing. Yeah, I think those are the risk factors.
Yeah, thank you for answering that. And actually, Sarah will also dive a bit deeper or actually kind of zoom out and talk more about foodborne pathogens in general and some risk factors associated with that. So we will hear more about that. Mm-hmm. So if there are no more questions, I don't see any more hands or questions in the chat. Then we will take a break now. And we will start again in 10 minutes with the presentation by Sarah and then have her Q&A. So I will put the time in the chat as well. For Sarah and some other people, it's still very early. So the time depends, but it's in 10 minutes we start again. So enjoy your break and get something to drink and see you in a bit. Welcome back, everyone. I hope you had a nice break. Yeah, it was lovely to hear about Akbar's background about schistosomiasis and parasitology. And now we will switch to Sara Garcia, who will explain more about the context of food safety and security and her own work. So looking forward to your presentation, and go ahead when you're ready.
Great, thank you for having me. Did Benny have something to present before I go? I just wanted to make sure.
Yeah, I think Benny had a short introduction. I see him on screen as well. I had a few slides of him, so I can start with the. Oh, yeah, he unmuted as well. Can I share the slides? Is that okay? Okay, then I will start with Benny's presentation. I will open the slides now. I think you should be able to see them now. So whenever you're ready. I think we cannot hear you yet.
Is it better now?
Yeah, we can hear you.
Okay, thank you. Thank you very much for the opportunity to be with you today and to give you a little background about the Western Institute for Food Safety and Security. It actually came into being about 25 years ago, mainly because California has been, in a sense, the salad bowl of America and other parts of the world. And there was increased interest in the fact that there were foodborne illnesses being associated with produce. In other words, vegetables, leafy greens, things of that nature. And so the Department of California Department of Food and Agriculture, as well as Department of Public Health, joined with the veterinary school to begin to put together a program to really find out what was going on with these foodborne outbreaks associated with vegetables and the like. So with that, funding came from the state originally to begin to look into this, and it was later picked up, as will be shown by Dr. Garcia, by the fact that the Food and Drug Administration was very concerned about what was taking place. So with that, we initiated the program. We actually had some individuals from China come by, and they liked the idea that, gee, this fit what we were launching as a One Health effort to look at the role of animals, plants, people, and the environment as a means of getting a better understanding of this cycle of foodborne pathogens, which we had always associated with animals and people to some extent in the past. So with that, as we started moving forward, we found that probably one of the easiest things to do is to try to interest capacity building by working with undergraduate students. And so we established a program with individuals, universities in Southeast Asia to begin to look at this particular cycle, both from our experiences in California as well as what may be taking place in other parts of the world. So what we did is we set up conferences, in a sense, courses that we provided twice a year, once in the summer and also during their winter break. And over that time, we began to demonstrate that it was important that we bring together individuals from a variety of disciplines that may be involved with the environment or with plants or with animals or public health, or that interfaces with people. So as we moved through, our main way of trying to raise the interest of students and the like would be to bring different faculty from on the campus together to help provide some lectures, interdisciplinary type introductions. And through that, we wanted to raise awareness of the breadth of the different disciplines that were involved in the One Health concept. And so one of the principles was to raise awareness. And the awareness was for selected topics to be given to teams of students. And next slide, please. And so we would put together to build team building exercises from veterinary students, animal science students, plant science, soil scientists, public health, and allow these teams to begin to look at a problem and together work on solving the problem. So this became an interesting means of directing the students' attention to a problem rather than sticking with a single discipline. And it was amazing what they would come up with as they eventually began to work together. And through this, they would put together a plan to address the problem, to come from their different backgrounds and indicate how they could come together to form a team that would be able to, if I could have the next slide, please, call to action. And it's been fascinating to see how this has brought together teams of individuals that they then had and began to understand the huge opportunity that they could have if they decided they wanted to continue on into post-discipline graduation from universities. And it's been a rousing success because we've had students from over 21 different universities in Southeast Asia, plus the fact that they have know each other, they have built bridges between each other, and a significant number have gone on for advanced degree programs throughout universities in the world. There have been some going to Wageningen, we have them at UC Davis, but they're also Europe and Australia, New Zealand. So we think that this program is one in which it really looks at the breadth, as Dr. Garcia will demonstrate to you in a few minutes. We're very pleased that Dr. Garcia, who has been working with WIF over the course of a good many years, and she's had some broad experience both with plant agriculture as well as animal agriculture in this regard. And her current position, which she just recently took, is an innovative approach, I think, for governments. But she's just recently took the position of Technical Assistance Coordinator for the California Department of Food and Agriculture. And this is to focus and really bringing into play the importance of environmental farming and innovation. And she's been, while she was with us at WIF, she had started moving into this area some time ago. And I think she will go ahead and give you a better background. But again, thank you for the opportunity. It was a fascinating talk on schistosomiasis, and I really appreciate that. But with that, I'd like to turn it over to Dr. Garcia. Thank you.
Yeah, thank you for the introduction. We're ready for the presentation.
Great. Thank you guys for having me. So I just like to kind of talk about my journey into One Health. I actually am from an agricultural community in Central California, which is very rural, generally like low education level, and most people work in agricultural day labor. And so my goal in life was to become a geneticist. And I came to UC Davis to study genetics. And that's where I met Dr. Osborne and started working at WIFs as an undergraduate. During my time I was studying, and after that I worked in mouse genetic analysis, where we created transgenic models of human diseases for biomedical researchers. So I did that for a little bit, and then I came back to do my PhD with Benny and Jim Culler in integrative pathobiology at the vet school, where I worked in both produce food safety as well as dairy production medicine and dairy food safety. And during that time, we worked in Rwanda supporting smallholder farmers with mastitis control and building capacity in Rwanda to diagnose the different pathogens that cause mastitis. We did a lot of milk laboratories, capacity building and training. And then for the last 6 years, I've been a faculty researcher at UC Davis, continuing that work, as well as working with Dr. Osborne on these education and training programs to build capacity, both internationally as well as here in California for small farmers that need help with these practices in agriculture. And so, yeah, I joined CDFA about 4 months ago. This is a new role that I joined. minutes. Okay, now I'm having a different problem. Okay. And so I'm going to talk about that in a little bit. But if we zoom out and talk about the global challenges we're facing that need a One Health approach, they're pretty broad. Globally, we know that our human population is growing really fast. We're expecting to reach almost 10 billion people by the year 2050. And along with this population growth, we really need to understand and find ways to address food security, address poverty with poverty alleviation policies and frameworks, and then also the conservation of our natural resources to preserve those wild areas, as well as our working lands, such as places that we have animals for food production or grow crops for food as well. And this really needs to consider all of the different interactions with the environment, how diseases are spread through food and water or by other vectors, as well as, overall improving livelihoods for people everywhere. So I just want to talk for 1 minute about what I'm doing right now at CDFA. So we have a legislative mandate to provide incentive programs to farmers. And those incentive programs help farmers reduce the amount of water. So we have a program called the State Water Efficiency Enhancement Program, and that helps reduce water for irrigating crops, as well as reduce the amount of energy that is being used to pump that water from the aquifer using ground pumps. And so this helps reduce greenhouse gas emissions and energy use, as well as conserve water. We have another program for soil health and carbon sequestration that provides ecosystem benefits. So practices like cover cropping, compost application, practices that really maintain or build the integrity of soil, as well as prevent nitrates from leaching into the aquifer, and other ecosystem benefits, such as like pollinator habitats and things like that. And then we have another program to reduce methane from dairy operations that really entails changing the way we're managing manure at these facilities, such that it reduces methane emissions. And part of all of this is the requirement to provide technical assistance and capacity building, both for people that do the work to help farmers implement these programs, as well as supporting the farmers in understanding why it's important to do these programs, helping them apply for the grant funding to be awarded and be able to implement these practices, as well as building capacity among our statewide network of providers that provide the support directly to farmers on a one-on-one basis. And this is something that I've been kind of getting into the last few years, and this position's really helped me see firsthand where there's gaps in training. And for these programs broadly, there's really amazing staff that are very knowledgeable about water efficiency, nutrient management with irrigation, soil health and biodiversity, as well as manure waste management. But from my perspective, there's really a big gap on this concept of One Health, as well as food safety and food systems. And that's where I see we can improve knowledge. So I kind of like to give this background about One Health at UC Davis. Dr. Calvin Schwabe was a UC Davis professor in epidemiology, and he wrote this book called Veterinary Medicine and Human Health. And in this book, he termed this coin One Medicine. And fundamentally, what he noticed through his extensive travels and readings and studies about health and public health, and he was very interested in people's cultural and native foods and how they prepared those foods and risk factors for zoonotic transmission of pathogens in that aspect. And through all of his exploring and studies, he basically noted that the disease in humans and animals and the treatment of that disease is essentially and fundamentally the same, and that there is really only one medicine. And so he really was early to acknowledge that comparative medicine was really important to improve the health of both humans and animals. And in his concept of one medicine, he saw that human health, animal health, and science were integral to achieving one medicine. And this also included social and cultural sciences, as well as the traditional bench and life science type of research. But his 3 pillars of one medicine were essentially that humans and animals both need access to safe and hygienic and healthy, nutritious food, as well as hygienic water. So water quality is very important. And then hygienic living conditions. And if we could achieve these 3 pillars, that we could improve the health of both humans and animals, as well as reduce the transmission of foodborne and zoonotic disease. So a little bit about food safety, because that's what we're going to talk about next. But there's 4 different types of food safety hazards. They're microbial, chemical, physical, and allergenic. Today, we're going to focus more on microbial, but a couple examples of these others include chemical. You might be familiar with aflatoxin outbreaks in dairy animals. These are typically caused by improper storage of grains that become contaminated with mold, and then these aflatoxins are present in the grain that is consumed by an animal, and that can be shed in the milk. And so here's a nice little image of that to show that cycle of transmission of a chemical through the food system, and we might inadvertently be drinking milk. This is a more common problem in lower income countries as opposed to higher income countries, and it's very common, something I saw commonly during my work in Rwanda for the time I was there. Another example of that is cadmium in rice. Cadmium can be uptaken through the root of the rice plant and located into the grain as the plant is developing, and this can be consumed by people. And over time, consumption of this contaminated rice can end up with serious medical effects such as osteoarthritis, kidney disease, and other things that reduce quality of life and health. Other common hazards that happen in the US are contamination of processed foods with metal and plastic. This is pretty common. And then allergenic contaminations of food. So people with food allergies could inadvertently eat food that has been processed in a facility where there were also peanuts or milk, eggs, or wheat, and cross-contamination could happen in that way. So those are 4 food safety hazards. So we're going to talk about microbial food safety for a little while as it relates to produce. So symptoms of foodborne illness include diarrhea, vomiting, abdominal cramps, and fever and lethargy. These are usually the acute symptoms, the ones that happen pretty quickly in the early stages of infection. And specifically E. coli 0157H7, which is a common contaminant in leafy greens, can lead to a life-threatening illness called hemolytic uremic syndrome, which is basically damage to the kidneys and kidney dysfunction. And then other pathogens that are transmitted through food, such as Brucella, Coxiella, Mycobacteria, Trichinella, can lead to more chronic types of foodborne illness that result in like general malaise, like recurring undulant fever, osteoarthritis, and chronic malabsorption types of syndromes, as well as causing kidney disease and other types of depression and chronic fatigue syndrome. So these are very serious health issues that can be caused by foodborne illness. So the 9 most important foodborne pathogens typically that we see here in the US are Salmonella, Campylobacter, Cryptosporidium, E. coli 0157H7, Clostridium, Listeria monocytogenes, Toxoplasma, Yersinia, and norovirus. And this slide here is to demonstrate that 8 out of 9 of these are derived from animals, and fundamentally, they're zoonotic and transmitted through food. And norovirus is the one that we recognize that as humans are the natural reservoir for this virus. But there's really a lot of research to show that there are strains of norovirus that naturally infect other animals, such as swine and cattle. But this is also, I would classify this as potentially a zoonotic disease as well, but there needs to be a lot more research done in this area. So we're going to talk mostly about E. coli 0157H7, as this is the main culprit in outbreaks of leafy greens here in the US. And I just want to talk briefly about the fact there's different types or categories of pathogenic E. coli. And on a big picture scale, these different types of E. coli, whether they're the type that adhere to the surface of an epithelial cell in your GI tract, or that they can internalize and migrate to other cells, this is determined by the specific set of genes and virulence factors that they carry in their genome. And this is a really important concept, because what we're finding is that these genes determine like the severity of disease, as well as the ability of these to persist both in humans and plants as well, which I'll talk about later. And I also want to really convey that for these pathogens, it really does not take very many cells to cause disease or illness. So in the case of E. coli 0157H7, less than 50 cells can cause disease. And depending on a person's health status, this could be a mild disease or a very serious life-threatening disease that could lead to death. But if you look at some of these, the infectious dose is really quite low for viruses. I know for norovirus, it's about 5 to 10 viral particles can cause disease and result in severe diarrhea. So I just kind of want you to keep that in the back of your mind as we move through this. Again, who's at the highest risk of experiencing foodborne illness? It's basically people that have some status of immune compromised health status. So cancer patients, children under 5 that don't usually have a fully developed immune system, diabetes patients, HIV/AIDS patients, older adults, people with autoimmune disease, and pregnant women. And so these pathogens are much, much more risky for these groups. So in the United States, foodborne illness affects about 48 million people every year and results in about 3,000 deaths. And that's really underreported, because many people might experience a bout of foodborne illness, but it might not be severe enough that they seek medical attention. So we really think that this is much, much higher than what is documented in our statistics. And if we look into the types of food that sicken people, you can kind of see like the big categories. Fish, a lot of people eat sushi here in the US, and that's typically raw fish that could cause illness. But also like improperly storing fish or other meat products at the improper temperature could result in increase of pathogenic bacteria if they're present. But if we look at this, it's kind of distributed in a way that here, basically from here to here is vegetables, right? And then here's some meat products, and then here's some nuts and seeds over here. And then, so just a big picture of the types of products we associate with foodborne illness outbreaks. But if we look at this table, it's much more clear to see that in the US, there's about, if we look at animals versus plants in terms of causative food products linked to outbreaks, plants account for about 5 million illnesses every year, or half of the outbreaks. And it's actually quite a bit more than animals. And when we break that down, produce, and the subcategory of produce, leafy greens are the highest cause of foodborne illness for a specific type of food commodity. And this is really not well known to the public that produce is the #1 cause of foodborne illness in the US. So what makes leafy greens risky? Well, many enteric zoonotic pathogens have a low infectious dose, right? We looked at that table, and most people are not really aware of that. And then it's not uncommon that the consumer, or maybe in transportation through the food system, that these products are not properly held at the appropriate temperature during that time that they're traveling to the market, or also that they're being consumed past the time that they're really fresh, right? And that's providing more time for bacteria to grow if those products are contaminated. And then there's a risk for amplification of these bacteria or cross-contamination during the process of leaving the field where they're harvested to transporting to the market where they're purchased. So there's processes here where different batches of leafy greens are washed in the same types of machinery. So if there is contaminated produce going through this machinery at the same time, it's basically cross-contaminating the other produce that's being commingled with it. And then again, produce is typically consumed raw or minimally processed. It's very common here for people to purchase and consume pre-cut, packaged, bagged lettuce. These have a higher incident of associations with outbreaks. And again, there's not a step of cooking or inactivation to kill these pathogens that might be present on the produce. And then something that's really, really not well known is that these pathogens, and I'm going to talk about E. coli again in a second, have the ability to attach to the surface of leafy greens as well as internalize into the edible parts of the plant through the leaves as well as the roots, and they can also internalize into the seeds and seedlings. And again, these are products that are consumed raw, basically. Meat, eggs, and milk are typically better known to the public about the cooking temperature, or buying pasteurized milk, although unpasteurized raw milk is more popular now, which is leading to higher or more occurrences of foodborne disease. And people are more well educated about handling meat, eggs, and milk because they know the improper handling can result in outbreaks. So I have a couple slides here. I kind of just want to go back to this idea of internalization into leafy greens by pathogens. So E. coli 0157 is what we're going to look at right here in the next few slides. This is an electron microscope scanning image of an experiment where they basically took seedlings of lettuce, I believe it was romaine. They planted the romaine and they, as a seed, and they irrigated it with contaminated water. So it was water combined with untreated manure effluent, manure water, basically. And they followed the E. coli 0157H7 labeled with a green fluorescent protein. So you can see here in green. And then each of these images, 0, 1, 2, 3, 4, 5, is 1 micrometer deeper into the lettuce seedling tissue. So this is the surface, 1 micrometer, 2, 3, and you can see that these E. coli have internalized into this lettuce as it was growing and is in the tissue. And this is really not well known to the public that pathogens have the ability to do this. So in this experiment, this is also E. coli that was inoculated onto the leaves of spinach. Here is the untreated control. So this green is the stomata, which is the little pore on the surface of the leaf where gas, oxygen, and carbon dioxide are exchanged as part of the respiration of the plant. The blue are the bacteria of E. coli 0157H7, STEC is another name for that. And the red is the flagella of these bacteria, which kind of give them the ability to latch onto things or be modal a little bit. And so what you can see is these are the start, 0 hours, 2 hours, 4 hours, 6 hours after inoculation with these 0157H7 E. coli, and that these bacteria basically start moving and congregating towards the stomata. They begin to internalize into the open space inside the stomata, and that they can enter the vascular tissue of the plant through this means, and that these flagella help them do that in adhering and internalizing. So this is actually an experiment I worked on with Benny at WIFS when I was working in the laboratory. We wanted to replicate vacuum cooling. And this is a process where they rapidly cool trucks filled with lettuce to bring them to the proper holding temperature. And they enter a vacuum chamber and the vacuum's turned on, and this removes, this basically cools the product through reducing the pressure. So here is the untreated, here is our control. It's inoculated romaine leaves, and these green dots are basically E. coli that have congregated on the stomata. And this is before we turned on our vacuum. So you can see again, naturally, these bacteria seem to move towards the stomata on the surface of the plant. After we applied the vacuum treatment, these STEC E. coli were internalized into the tissue of the romaine lettuce. So this is another issue with leafy greens that's not really well known by the public. And so just kind of keep that in the back of your mind. We're going to talk about animal health, food safety, and food security. So let me go back to the produce food safety for a second. The losses due to outbreaks are pretty big to producers, so they want to prevent outbreaks of foodborne illness, and prevention is really the key. Preventing the contamination of these products on the farm is really the most important thing we can do by implementing on-farm food safety practices. And in that context, we're going to talk about animal health in the big picture next. So animal diseases are also very costly to producers. The treatment of these livestock, the losses of livestock due to death, and some diseases that the food animals carry can also cause occupational risks and are zoonotic and can cause health issues with people that are working with or handling those animals. And again, as we saw with lettuce, there are some pathogens that can enter the food supply chain. The example of consuming unpasteurized milk is a really great example of that. And then also these pathogens can be transmitted and contaminate the environment through water, wind, or dust, as well as contaminating other commodities if there's a produce facility next to dairy. That's a cause for concern. And when there are outbreaks of catastrophic disease, such as high-path avian influenza, this really impacts food security by limiting the supply of available animal-based proteins. It results in increased prices for consumers, as well as food recalls, and ultimately resulting in food insecurity. So some animal diseases of concern include Mycoplasma bovis in dairy cattle, Brucella abortus, and some of these are also endemic in parts of the world, and we've largely eradicated like Brucella and Mycobacterium here in North America, as well as foot and mouth disease. So these are diseases that we're concerned about here in the US and want to prevent from entering our continent. And a really good example of another one is in poultry, highly pathogenic avian influenza. We've had a multi-year ongoing outbreak of this, which I'm going to talk about in a second. And then others related to swine include classical swine fever and African swine fever. So these are all diseases that are high cost to producers, or if they are to enter the US, are going to devastate our food system. So I want to talk about highly pathogenic avian influenza. So this is classified as a foreign animal disease here in the United States. So this means when there is an outbreak of avian influenza in poultry facilities, action is taken immediately to contain and mitigate and basically remove that threat from the area so it doesn't spread to other producers. So in 2014 and 2015, we had an outbreak of avian influenza. Basically 50 million birds were affected. What does that mean? Affected means that those birds, the facilities where those birds were located, were basically quarantined and the birds were destroyed. So they were euthanized. And this is a huge loss to farmers and a huge problem with the availability of these food products. So typically we see increased prices when this happens. And this is a lot of animals. And we really want to think about prevention in terms of how do we prevent these infectious diseases from entering the food animal production system. In the current outbreak, it started in 2022. It's still ongoing. More than 100 million birds have been affected across 48 states. And you can see, roughly half of our states were affected in 2014-15 outbreak. And now almost every state is affected with this current outbreak. And it's estimated that the losses are at least $3 billion in direct losses, double what that loss was for the 2014-2015 outbreak. And really the change of migration of these birds, the increase of climactic events is increasing the incidence of these types of outbreaks. And this one is really unprecedented because it's been going on so long. Globally, many different types of mammals have been infected with this avian influenza strain that is going around. All different kinds of mammals, many birds, as I just discussed. And now here in the US, early in the year in January, it was detected in dairy cattle. And this has been a wake-up call for dairy producers in terms of biosecurity and protecting their herd from zoonotic infectious diseases that can transmit to different species, not just within the same species. And there are, again, this is devastating wildlife all around the world. And just to kind of drive that point home, this is an image from the USDA where they are tracking the H5N1, that's the strain of avian influenza that is implicated in this outbreak. These are all the different types of mammals that they have detected this strain of avian influenza in the US. And as you can see, it's very diverse, from cats to bears to seals, coyotes, foxes, mink, otters, squirrels, house mice, raccoons, skunks, and possums. This is almost every different kind of wild animal that we have in the United States. And so, there is a big concern of these diseases passing from wildlife to food production animals, as well as to humans and vice versa. So this year there have been about 7 people who've contracted H5N1 either from a dairy facility or a poultry facility. So this is a very serious concept of preventing transmission of zoonotic diseases. So when we talk about prevention, what we're really talking about is biosecurity. What kind of biosecurity do we need to implement both at produce farms to prevent contamination of fresh produce, as well as at food production facilities to keep our herds and flocks safe from these catastrophic diseases. And that kind of starts with understanding how diseases are transmitted by people, animals, as well as insects. So these diseases can be transmitted by wildlife, by rodents, insects. They can blow away in the wind on dust particles. They can enter the water supply if water is contaminated and going into a surface water supply and being maybe down the irrigation or canal being used for irrigation on produce. Employees and visitors could introduce these pathogens to a facility on their shoes, on their vehicles, maybe on their clothes. So these are all things that are very important to consider, as well as providing hygiene for people that are working. Where do they go use the restroom? Can they wash their hands? Where can they eat their lunch away from animals or the produce production lots where they might be harvesting lettuce? And then another big consideration is manure and litter management. So one of the programs we have at CDFA is helping farmers find alternative ways to manage their manure to reduce greenhouse gas emissions. But I think we should also include training on how do we manage that in a way that prevents the transmission of pathogens that might be present in that untreated manure. What are different ways we can compost or treat these waste products such that these pathogens are inactivated and can re-enter the food system in a sustainable way as compost, which there's a lot of research on that area as well. And then again, transmission by vehicles and equipment, and how do we keep those vehicles and equipment clean to prevent transmission of disease or pathogens. So really what we're talking about on a big picture scale, we first started at the farm with produce and food animal facilities, but thinking about that as a food system, how might these pathogens transmit from the farm and go through our food system or enter the environment to potentially contaminate other areas or products on the farm through the food system, at the market, vehicles, and in a processing facility. So a big picture understanding of this framework is really, really important. And then I just have one little slide here I like to use in an activity that Dr. Osborne was talking about at our conferences. This is kind of might be a very typical scenario here in California where a produce farm might be located next to an open air poultry operation. This is some manure lagoons next to some running water. And here's some beef cattle up on a hill where potentially their waste when it rains could go downhill, or the wind could blow contaminated dust into an area where we might not want it to land. So this is just to kind of put it in a on-farm scenario that people can think through and talk about what are those risks? How might we prevent transmission of those pathogens in this type of setting? And so with that, I'm going to end with just talking about One Health and food systems, and that really we know a lot of people really recognize that One Health is the health of humans, animals, and the environment is interconnected. But more and more, finally, people are really recognizing that plants should have their own space in this One Health paradigm, that plants are vitally important in the food system and for ecosystem health benefits. And we really need a broader concept of what One Health is that incorporates the food system, veterinary medicine, human medicine, environmental health, and acknowledging that food and agriculture are tied to public health, livelihoods, and social equity. So with that, I'll stop. Thank you. Do we have any questions?
Yeah, thank you for your talk. It was really interesting to hear about the high risk of like these plants or leafy vegetables for food safety issues, even though like normally indeed animal products are typically seen as more risky by the public. And the public includes me as well in this case. So yeah, it was really interesting. And also that your view on the risks associated with the food itself, but also the risks of the current animal production system. So thank you for your talk. And we already have a question in the chat by Tiken. What are the social cultural determinants that affect food safety and security in low and middle income countries? And what are the mitigation strategies to be taken to tackle the challenges?
Yeah, I can speak to that from my experience in travels in Rwanda as well as in China. Really, in low and middle income countries, poverty or low incomes really prevents people from being able to access and afford clean water and clean food. And culturally, there are, ways that food are prepared that might not inactivate foodborne pathogens that are present in that food. So fermenting food typically reduces the presence of those pathogens, but there are some, such as Clostridium, that can survive canning or fermenting. And so lack of awareness around properly preparing food for the purposes of food safety, I think are still really big challenges. And then again, because of poverty, there are still people that will enter the wild areas to search for animal proteins such as bushmeat. And, again, a lot of that, some of that is cultural, but I do feel a lot of it is not being able to afford food from a local market that was grown in a more sustainable way. And again, education and awareness around the risks as well as the impacts on wildlife and wild areas by those activities.
Yeah, so like the key aspects remain very essential still to kind of mitigate the risks over there. Okay. And then someone agrees with the space for plants and maybe always also soil and water. So the circle keeps expanding. Yeah.
Yeah.
It almost needs branches.
Absolutely.
And then another question by Jackie. So you mentioned that aflatoxin significantly impacts also these low and middle income countries, likely due to the high costs of food preservation, leading people to rely on available resources. And what are the best practices to adopt to minimize post-harvest food loss and foodborne illnesses?
Yeah, so I could speak to my experiences in Rwanda. Basically in the global south, these are very tropical and humid environments, and really preventing these grains from getting rained on or getting wet and reducing the humidity in which they're stored is really difficult. One, because of the amount of rain or humidity, as well as to the infrastructure. In Rwanda, a lot of the grain is stored out in an open covered area, but it's not enclosed in like a climate or temperature controlled silo or anything like that. And it at this point, it's really not practical economically to do that. So this, again, economics remains a really big part of improving the food system and making sure that the food supply is safe and secure, not just in higher income countries, but also in middle and low income countries. Oh, you're on mute.
Thanks. Thank you for your answer. I hope that that answers the question from Jackie. And I also see a hand by Fatima. So feel free to unmute Fatima and go ahead.
Thank you very much for your presentation. I really enjoyed your presentation. And then I have a quick one. Mitchell talked about, the public's focus being on foodborne pathogens from animals mostly. But then It's time we all pay attention to foodborne pathogens from the vegetables and the leaves. I saw Cryptosporidium among the 9 pathogens or 10 pathogens that you mentioned that are the key causal agents of foodborne diseases. And I saw Cryptosporidium among them. What I know is Cryptosporidium can be transmitted or can, it's normally, it's a zoonotic disease, meaning it can be found in humans, animals, and the environment. But one new thing that we are seeing here is Cryptosporidium being a foodborne pathogen, meaning we can find it on the leaves too. And in my question, simple question is, why has crypto received less attention in its, potential of being, a foodborne pathogen in the area of the leaves and the vegetables? Thank you.
Yeah, that is a fantastic question. And I think Benny could chime in on this as well. Our group at WIFS has been studying this for almost 20 years, and it's still very understudied. And there is still the idea broadly, even among researchers, that pathogens that are sourced from humans and animals, those are their main hosts, and that they don't enter the plant system, basically. Even though we know that E. coli is implicated in produce. And this kind of comes back to the idea that people are not aware of the ability of these pathogens like E. coli to internalize into the tissues of plants, or be uptaken through the roots, or be internalized into the seed. And so people think it's more of a contamination issue, like with water sources, which it can be, but that still doesn't address the fact that of internalization, right? People think they can wash their lettuce and they're going to wash off the E. coli that could potentially be on the lettuce, but that's not actually how the system works, because those bacteria can internalize into the stomata. And the other thing is, I just think overall, we're not studying these pathogens with this lens broadly in food safety. So people are looking at it more from a zoonotic water contamination or water quality issue, as opposed to what is the actual molecular interaction of these pathogens, bacteria or parasites, with the tissues or the surfaces of these produce products, because there is good research to show that, like, E. coli has to have the flagella system or the type 3 secretion system to be able to adhere to the surface of leafy greens. So at a molecular level, we need to really understand this process a lot better, as opposed to thinking about, oh, well, the E. coli got there because of the contaminated water, but we can just wash it off, right?
Okay, thank you. Yes, the reason why I'm asking is my group here, we started some work on Cryptosporidium in Ghana here. And, I'm working in the northern part of Ghana. And in the northern part of Ghana, due to climate change, there is shortage of water, but we have a lot of artificial dams here. So people tend to depend on these dams for agricultural irrigation, swimming, and others. So we went to some of these dams to take samples, and we have farms around these dams. That's the dams irrigate those farms, but it didn't click our mind to also take samples from the dam to check. So I'm very intrigued by this new thing, because we picked water samples, looked at the human and the animal. We are still on it. And it's interesting to know that we have to also look at the plant or the farm products or the produce. Thank you.
Yeah, you're very welcome. Yeah, water quality, again, the 3 pillars: water quality, hygienic food, and hygienic living. But really also, I think, understanding what the molecular interaction is between, bacteria and plants, in the same way that we study it in the context of host pathogen interaction in people or animals, I think is really the next step to being able to find solutions for these problems.
Yeah, thanks for giving more background information about how the E. coli transmission works and how it can like get into the stoma as well. So I think we can do one more question. It's a short, like for a short question, maybe it's hard to answer it in a minute or so, but Lencho says it is an interesting presentation. And what do you face while trying to implement One Health?
You know, I think in our space of veterinary medicine and human medicine, it's a concept that's been talked about and taught and being implemented, but broadly among the public, I think we need to have more engagement with the public. We need to learn how to be good scientific communicators, and how to build collaborations and connections to do multi-partner work in terms of policy and research and education. And that is really what implementing One Health is. I think you'll see my presentation's a little bit different than typically what others might present. And that's because I'm really trying to communicate the big picture of One Health, so that people can become intrigued by something where we really need a One Health approach, considering these complex systems, and being able to, you know, continue on in their journey of becoming a One Health practitioner with that lens.
Yeah, thank you. Very concise answers. So, yeah, I think this also kind of relates to how you implement the climate smart agriculture, like capacity building within your work. So I really liked how, listening to that. I will give a very short feedback session, like just 2 minutes, just to wrap up the session. And I want to really thank our speakers for today. So I will just share my slides. Okay, this should work. So yeah, for today's aim, we wanted to provide an overview of the One Health approaching food safety and security and examine the importance of interdisciplinary collaboration, so within One Health, and public education through case studies. So I think we did that well through the presentations by both Akbar and Sara. Akbar showing his PhD work, and Sara giving a more broad overview indeed, and explaining capacity building. So our next event will take place on August the 29th. It will be about livestock under One Health, and it will be a panel discussion type of format like last time. And we will have Amir Muse Osman. He's a PhD candidate at the University of Paraná in Brazil. And we will also have a speaker by Feo, which is we will announce that later. So I don't know if there are any last questions of today. Otherwise, I think we will wrap it up and call it a day. And I don't see any questions in the chat or hands, so I think it's okay. I hope to see you next time. And just one last thank you to our speakers, and bye-bye.
Bye.
Bye.
Thank you.
Thank you. Bye.
Thank you.